Our next presenter is Dr. Gianna Hammer, who I've also had the opportunity to meet at one of our V-Scholar Summits, another rising star who we think the world of. She too is looking at the microbiome, and in this case, what's going on in the intestinal tract. So without any more introduction, because she is the best one to tell you about her work, Dr. Hammer.
Good morning. I am Gianna Hammer. I'm at Duke University, and together with the V Foundation, we are battling against colorectal cancer. So the whole world appreciates our immune system for what it does. Now, it fights against foreign invaders. It fights against virus, it fights against bacteria, and as one of our most recent Nobel Prize laureates now determined, it also can fight against cancer. And in this dynamic, one immune cell is really key for this, and that's the dendritic cell. The dendritic cell's job is to detect foreign invaders. It can detect cancer, it can detect tumors. And what they do, they take this information and instruct immune responses. And the kind of immune cell that's key here to instruct is the T cell. The T cells are the executioners. They eradicate pathogens, then they can eradicate cancer. And T cells come in different flavors. And if you want the best kind of executioner, you want a T cell that makes a protein called interferon gamma. And the same kind of theory of hierarchy of who's better at the job goes also for the dendritic cell. They also come in different flavors. And the one that seems to be key for killing cancer is the one in red, which goes by the name DC1. So DC1 is known to be the champion against skin cancer, pretty well studied and well appreciated. It's also the champion against lung cancer. And our question has been then, is it the champion against colorectal cancer? And this is not simply a question of going down the checklist and asking this cancer yes, this cancer no. Colorectal cancer occurs in your gut, and your gut is so unique in that it's full of food, it's full of mucus, it's full of bacteria, fungus. All these environmental influences are unique to and just sitting right on top of the tumor and play a big role in helping the tumor grow, or maybe even also helping the tumor be eradicated. So we want to study this unique context of cancer and the role of dendritic cells in it. We do this using a mouse model. Mouse are helpful to us because they can also get cancers, and then it allows us to test there are certain roles of different immune cells. So this is a picture, an image of a mouse colon. And what you see by the white arrows are tumors. Those are spontaneous tumors that happen in this altered genetic background in this mouse. And what we have been asking then is looking at a normal mouse, a mouse that has all its different dendritic cells, and comparing the outcome of tumors from a mouse where we specifically take out our DC1, specifically take out the dendritic cell that we are thinking is going to be champion. And the idea here, of course, is that if you take out DC1, and if it's truly so important for fighting this battle against colorectal cancer, you would expect you take it out, and therefore you'll get more colon tumors. So what we first did is we take that tumor out and we ask whether or not the dendritic cells can even get inside. And this is an image of the different dendritic cells that enter the colon tumor really look like. So there are different flavors. You see some population here, another population here, but in this area is where we have the population of DC1. And we made a mouse where we take DC1 out, and sure enough, that population is missing. So our champion is gone, but the outcome was quite surprising. So the number of tumors in mice that have DC1 and the other dendritic cells was more than the mice that we took our potential champion out of. And we had several mice that actually has no tumors whatsoever. And when we quantify this tumor number, here are control. Control means has all the different, we did nothing else different. It has all its dendritic cells. And here's the mouse where we take that, our potential champion. And so again, it was a surprise to us because we thought this would be important in killing tumors in the colon. But if you look at the data says the number of mice that have no tumors whatsoever jumps up to 25%. And if we ask about mice that have 4 or more tumors, that number of mouse is very, very low if we take out DC1. So it surprises. It suggests that what we thought was going to be the champion is actually promoting colon tumors. So what we know about DC1 in skin and lung cancer is that it has this special relationship to make T cells that produce this protein interferon gamma. And those T cells are definitely anti-tumor. And coming now to the colon tumors, this unique environment where food and microbes and all these other foreign components can play a role, then we're asking, does DC1 actually have the same job? Does it make anti-tumor interferon producing T cells? And also, what could it be doing to promote tumors? So the first component of the question, it was straightforward to answer. So we just looked in the tumor and asked about T cells that produce interferon gamma. We quantified them. And so what we found actually was that the number of interferon gamma T cells in mice that have all the dendritic cells and mice that where we take the dendritic cell out, it actually was no difference. So it doesn't look like this DC1 is important for making the anti-tumor response, which is quite different than how it behaves in skin and lung cancer. So we turn our attention then to asking, what could it be doing to promote tumors in the colon? And here it becomes important to realize that T cells can come in at least two flavors. We know that the interferon gamma flavor is important for killing tumors. And there's another flavor that's really, really prevalent in the intestine. Actually, it's mostly lives in the intestine. And that's the kind of T cell that makes the protein called IL-17. We asked about how many IL-17 T cells can actually be in a tumor. And it turns out that a healthy colon has a moderate amount of IL-17 producing T cells. But inside the tumor, this number swells and it really just takes over the population there inside of the colon tumor. And prior research has really well documented that unfortunately, this cytokine, this protein, IL-17, causes colon tumors to grow. It makes them get bigger, and it even can cause some of the mutations that are driving cancer in the colon. So we asked whether or not we take out the DC1 and asked about changes on IL-17 itself. And sure enough, when we take out what we thought was going to be a champion, IL-17, which helps tumors grow, actually goes down. We asked about what T cells are then important for being controlled by DC1. And here's where we had another turn of events that also surprised us. So the relationship between DCs and T cells is mostly described for what we would consider a conventional T cell, which fall into this category. We call them CD4. And so those T cells do enter colon tumors, but they were not different between our control and mice that don't have a DC1. However, another flavor of T cell can make IL-17, and it's a weird T cell. It has a weird looking name. This symbol means it's a gamma delta T cell. It's a weird symbol. It's a weird T cell for reasons I'll go into in just a minute. But what seemed very clear was that the number of gamma delta T cells that can make IL-17 very clearly went down when we take out DC1, meaning that making a pretty seemingly clear relationship that what DC1 is doing inside of colon tumors to promote tumorigenesis and cause colon tumors to grow in colorectal cancer is expanding the number of these funny T cells called gamma delta T cells that make IL-17, gamma delta T17 cells. So through a set of experiments, we learned that when DC1 enters the colon tumor, so this is your intestine here, a cartoon representation of a tumor. When DC1 is inside there, it's putting out come hither signals, much like a very scary head coach calling you out and telling you, come here. This is how the immune system cooperates and listens to what dendritic cells say. So in this case, the dendritic cell is calling the gamma delta T17 to come hither. And we also know that it's causing those cells to proliferate rapidly inside that colon tumor. And this protein that these gamma delta T cells are making, IL-17, is just feeding tumor growth. And this come here signal we learned is actually so strong that it's attracting a crazy rule-breaking gamma delta T17 cell into the colon tumors. And what I mean by this rule-breaking gamma delta T cell are those that come from adult life, or non-fetal life. So gamma delta T cells are interesting and weird. For one reason is because they are made before you are born. They come during fetal life. They enter the intestine, again, in the fetus. They enter other environments where they know microbes are going to be needing to be controlled. And so they're there as sentinels to make sure that infections can be controlled. And again, they come during fetal life. And so once a person is born and also on into adulthood, no new gamma delta T17 cells come out. It's totally something that happens inside the womb. And what we learned was that colon tumors contain these rule-breaking adult origin gamma delta T17 cells. And we distinguished between the two and found that yes, in colon tumors, we do have the ones that are conventional, the ones that come through fetal life. But it seemed quite clear that while the healthy tissue didn't have these adult origin gamma delta T cell, colon tumors do. And when we took out DC1, what goes away are those adult origin gamma delta T17 cells, providing a really clear link, not only to this one T cell subset that's making the cytokine that cause tumors to grow, but also highlighting this weird population that really is breaking the rules and is expanding in colon tumors. And we know it's expanding because of DC1. So these adult origin gamma delta T cells and the DCs that expand them represent new therapeutic targets for colorectal cancer. And this is really exciting. And so therefore I borrowed a V Foundation's nice little slash here to highlight the outcome of research, the outcome of what can be discovered using mouse models and really dissecting out what individual cell types play, what do they do in colorectal cancer or in cancer in general. And so we're very excited about these findings. Here is my lab at one point of time fighting colorectal cancer. You can see we're on a crushed colorectal cancer run. And we were talking about the V Foundation to whoever would listen to us as we were running there. And we took nice pictures under our inflatable colon. And I definitely want to thank my student who we've been working together in this fight. And we've been thrilled with the V Foundation support. And yeah, he's very enthusiastic as I am about this work. Thank you so much, the V Foundation.
Thank you so much, Dr. Hammer. What a great presentation. And I bet everybody can understand now why it is that we love our V scholars so much.